Good morning, everybody. Welcome and thank you for joining us bright and early today on a Friday. Um, we'll get started right away here. Just give me a second to grab our slides. All right, so we've got an exciting presentation today, long awaited. We've got a handful of TEPIs here to share their experience and just their tips and tricks about publishing about Tiny Earth. My name is Trang Tran, and I am the Tiny Earth Outreach Specialist, and I help folks like you get connected to Tiny Earth resources, and I help host events like this as well. Feel free to introduce yourself in the chat. You can share your institution and maybe where your class is in the research flow or an exciting discovery from this semester, and we'll keep an eye on that too. We've got a dense agenda for today. So we've got 5 instructors who will be, like I said, sharing their tips and tricks about a specific topic about publishing. So we've got publishable topics and tips from an editor, piloting a study, all about garnering institutional buy-in and tangible tools, and selecting a journal. And then we'll finish off that content with non-traditional dissemination. And then at the end, we'll have a Q&A, so save your questions for then. If you want to, you can also put them in the chat and we'll address them at the end here. And with that, I will pass it off to Betsy Martinez-Foss.
Good morning, everyone. I hope you're having a wonderful day. And I think it's finally starting to feel like spring here in Minnesota. So for 10 years, I served as an editor for the Journal of Microbiology and Biology Education. I have been working with Tiny Earth for the past 4 years, and I am part of the Tiny Earth Science Committee, and I was the chair of the subcommittee that was trying to help TEPIs publish. So this is why I'm here, happy to share my experiences with you. Can I go to the next slide? So as you know, we all have experience publishing something during our graduate training, either multiple papers or perhaps a few, but most of our experiences come from publishing research, right? So we are very well at publishing research, but we are very good at publishing research results. Then suddenly when we go to an institution that is primarily undergraduate institution, where we are doing mostly research with students, and we also get involved in some research related to a scholarship of teaching and learning, then perhaps it becomes a little harder to figure out what's publishable. And the Tiny Earth program provides multiple opportunities to think about publishable items. The first one that I have here, let's see if I can get the laser pointer going. Okay. Oh yeah, here we go. Okay. So the very first one is if you have enough results that you have gotten, over multiple years and multiple courses, if you can think about publishing those results. And later in the webinar today, you will hear from Angelo, who did that, and from Adam as well. So that's one avenue, going into any traditional research journal and publishing research results. We can also think of publishing classroom activities. Many of us have done that. There are educational journals that welcome these type of activities. Also, recently, I have seen multiple QR communities that have gotten together to write a collection of review articles that are related to the topic that they study. For example, in our case, Tiny Earth, we are interested in antibiotic production. Every time I am working with my students, we cannot find so many comprehensive reviews about what organisms produce antibiotics and what is known about the mechanisms of antibiotic production in general. So another idea could be we can get together as a community, a couple of instructors, and write review articles that might be helpful not only to students, but also to other Tiny Earth instructors. And finally, we can also think about publishing materials about our experience in the Tiny Earth community. And this is something that other QR communities have done successfully. So CPhages and the malidehydrogenase QR community have papers that are like, a multi-year study about what the student learning and faculty experiences as part of those programs. Can I get the next slide? Okay, so I want to briefly tell you about some ideas for research that fit within the scope of Tiny Earth. So one of them is publishing draft genomes as part of a microbiology resource or a genome announcement. Okay. In addition to that, there are journals that welcome metagenome sequences and transcriptome data. So those are, ideas for short communications. They are usually anywhere from 500 to 1,000 words, but they're usually getting around very quickly. So if you are, thinking you need to have publications to apply for tenure or for a promotion, these are a really useful tool to use, the genome resource announcements. Then, of course, as I mentioned before, we can have research papers, either a full article or a short communication that is slightly longer, about 2,500 words. There are many, many journals that are happy to receive this type of papers. And then finally, as I mentioned, review articles. And these review articles can be about antibiotic production. It could be about a particular type of bacteria that we study and we're interested in. So can I get to the next slide, please? And then when it comes to teaching, there are a variety of pieces of information that we can publish as part of our classroom activities. So one of them could be laboratory exercises. Did you do something different with your Tiny Earth project that you want to tell other instructors about? Any classroom activity. And this one, if you go for these tips or ideas to explore, these are like what the sections are called in the education journals. For this, you don't need a lot of assessment. So if you have no assessment or if you only tried it once, it's fine because this is an idea to explore or this is a tips and tools. So it's just to let people know what you did and how it worked. It doesn't have to be extensively assessed. Now, you can also write a curriculum article. Those tend to be longer. They need often IRB approval and multiple rounds of assessment, okay? And finally, if you are writing as a community, then we can publish meta-analysis of review papers, and those are going to need multiple years of data collection, as well as very extensive statistics and data analysis. Next slide, please. So I want to finish my part of this webinar by telling you about some things that you need to keep in mind before you think of publishing anything. Well, the first one I always think about is, do I need IRB approval? And do I need assessment? So what's the scope of what I'm doing? And if it's educational, those are one of the first two things we need to keep in mind. Who are going to be my co-authors and my collaborators? And how do I define authorships? What are the rules of the game? Who gets to be an author and who doesn't? What is your timeline? Some journals are wonderful, like Life Science Education is wonderful. Well, their timeline could be up to a year. I mean, we just got a paper published there. It took almost a year to get it through. Can you wait that long? Is the other thing. In addition to that, keep in mind potential reviewers. It's always good to submit an article with some ideas for who can be a reviewer. That saves time, not only to you, but also to the editor. Publication costs. And there are a variety of journals. Can you click one more time, Tram? Thank you. So Angelo is going to talk a little bit about how to select a journal in his part of the webinar. But what I want you to consider is that there are multiple journals for education as well as for research. And, they are all welcoming of a student-driven research, especially at the undergraduate level. And that's all I have for you today. And next slide. It is a pleasure to introduce Adam Kleinschmidt from University of Dubuque.
Thank you so much, Betsy. So Betsy did a wonderful job of kind of laying the groundwork of, what's publishable and how to work towards that end. And so I want to pick up from there and talk about, how you might potentially pilot a study in a collaborative fashion. So to get things started here, I just want to have us kind of think about, you having a potential idea and how do you get things up and going to pilot a study. And so any of us, we may start out at different points along a continuum. So for example, perhaps you're implementing Tiny Earth and you have run into an instance where students are challenged by a specific topic or a technique. And so maybe you see a room for innovation, right? And so that might be one starting place. kind of on the other side of the continuum, perhaps you've already taken action and you've drafted a curriculum piece or a new research technique or adapted a research technique to fit into Tiny Earth context. And perhaps you just want help refining that, that technique or tool or testing across various types of classroom implementation context. A third potential approach would perhaps you have more of a conceptual question that you'd like to test or further tease out. It could be education in nature, or perhaps more classical wet lab-based biological question. But either way, it may be useful in many ways. It may be useful to take a collaborative approach. And this may be something as simple as, talking to another TEPI and using them as a sounding board to help refine your ideas and kind of tease out exactly what you want to do or the next steps for piloting. Or potentially you're doing something more active, right, where you're looking for collaborators to work on that project. And I find both these things are very useful. The latter helps me keep on track and meet deadlines and help those scholarship projects keep going. So there's some animation here. So here I'm going to suggest, how can we leverage the Tiny Earth network to help doing this? The network, as we know, continues to grow, and there's all kind of intellectual capital that TEPIs can harness. Next slide, please. So one approach to using a collaborative approach would be to generate a community of practice. And this is what I'm going to advocate for today. So what exactly is a community of practice? It's a group of individuals with a shared domain of interest. So perhaps TEPIs that are interested in antibiotics or cures that interacts and learns together over multiple meetings, right? So this is not like a one-off meeting. This is a group that meets at regular intervals for the period. The duration may depend on the goals of the group. And the idea is to document ideas and generate products, right? So you're actively trying to create some type of product to address the objective or the need. So in the bottom right, we can see there's a Venn diagram that really takes the three core components of a community of practice and kind of shows how they meld together. So again, individuals from a specific community, they're all interested in some domain of knowledge or research and working to create some type of product. So these community practices can really look how you would like them to look and work depending, again, on your context. But I do have some suggested guiding principles here. And I'll just highlight a couple of them. So the first one here, determining what those shared goals and commitments are going to be for the community of practice. And this may seem obvious, people are coming together to work on some specific problem. It may kind of seem obvious, but it may often is useful to spend a session or two really diving deep into exactly like what those goals are going to be and make sure everyone has full buy-in because community practices can be a commitment. But again, the payoff is the product. Heterogeneity is often a good thing as well. And we can think about this from many different contexts as well. So perhaps experienced TEPIs versus maybe a first-year TEPI have very different perceptions. And that can be very useful, especially for curriculum development. So how might you start a Tiny Earth community of practice? So one way to get started is to reach out to some of the Tiny Earth leadership, and they may be able to help you connect with TEPIs that, again, may be able to directly help you or identify further TEPIs that may be of interest for the community of practice you're looking to generate, and or help you put out an announcement within the Tiny Earth newsletter to, again, recruit TEPIs to potentially join your community of practice. So with that, let me just give an example of what this might actually look like. So I've had a chance to develop a supplemental learning module for Tiny Earth that's readily available. It's called the Bioprospecting module. And myself and some other TEPIs have gone together in a community of practice over the past year and a half or so here. And our major objective, overarching objective, was to publish this pedagogical module along with some supporting assessment data. And so last year, our sub-aim was to develop a pre-post learning gain-based assessment tool and obtain IRB approval. So that's something we accomplished last year. And then this year, we've had a chance to actually implement the module in the classroom along with data collection. This includes individuals within the community of practice, as well as other fantastic TEPIs outside of the COP. that have been willing to implement and collect data in their classrooms. And so here you can see some of our preliminary data from fall, which is encouraging, suggesting students are learning something from the module. And so we've also collected some data this spring as we move forward towards eventually publishing this in an educational journal. So with that, I'm going to transition us to Brian Merkel, who is a TEPI and faculty member at University of Wisconsin, Green Bay, and he's going to talk to us a little bit about garnering institutional buy-in.
Thanks so much, Adam. It's really good to be with all of you. So next slide, please. I will get to institutional buy-in in a moment, but I really think I need to start here for several reasons. So this really ends up being, I think, a cautionary tale of how serious this can be if we're disconnected, if we're not assertive, if we're not mindful of some of the things that I'm going to mention that relates to my own department, of which I'm chairing now. So a couple things about this department, human biology at UW Green Bay. We have lost over the last 20 years 3 cell biologists. It has everything to do with their perceived lack of scholarship at our institution. So, you know, there's really a cost here, right? Human capital. We don't have any full professors. That's likely to change. And I am actually the first person to go through the firewall, if you will, if you want to think about it this way. And a big part of the reason why I'm at a point now where I'm going to be in a position to do this, quite frankly, is because of Tiny Earth. So UW Green Bay, we have a 21 credit teaching load to provide some context. 3rd most popular major. So we advise a lot of students, overwhelmed by students in many cases. Many of us do biomedical research, right? This kind of gets into what Betsy talked about, right? And I call this the R1 mindset dilemma. And I think it's very difficult in my experience for a lot of individuals psychologically after they go through a process and getting their PhD. And I've seen it with these cell biologists specifically. It's very difficult for them to think, well, is this really worthy what I'm doing now or what I'm trying to do? Because I know what I used to do. And no matter what, I just have to keep trying to do that. Even though I'm here and there's 21 credit teaching load and I have a lot of students I need to advise. And even though we don't have a baseline budget, somehow I have to figure out how to get funds to purchase consumables. And I mentioned the mindset, the R1 mindset dilemma. Students at our institution in our program need research experience to be competitive. So it's almost like an unwritten rule for a lot of med schools that unless students are doing research at the undergraduate level, it's something that ultimately is really important. So while we enjoy working with students, it's not like you're working with a postdoc or a graduate student, right? And so if you're doing cell biology research or immunology research, what have you, with all these kinds of things, this is a challenging situation. So while I think we can say there's a multitude of reasons why we've lost 3 cell biologists over that great span of time, which comes at a great cost to our program, to our students, to the institution, the people that have gone through this, which have been impacted most directly and severely by this. I would say failed mentorship, maybe even, but there are a lot of reasons, right? So I'm highlighting some of those that really kind of connect to the theme of this webinar. So if I could go to the next slide. So suggestions, and I'll get into the garnering institutional buy-in, right? That's gonna be the last thing I talk about, maybe for 30 seconds, but maybe at some point we could do a deeper dive on that and what that looks like. But it's really critical to adjust R1 thinking. And I think we can throw this up on a slide and it would appear otherwise to be something simple to do. I have just seen it over and over and over again, that it is extraordinarily difficult for some people to really let go of that. Redefine your scholarly activity, things that we've talked about here, others have talked about here, journal choices, all those kinds of things. Collaboration is so important. It is the Tiny Earth way, right? That's one of the greatest strengths about this network, starting with people like Jo Handelsman, right? She's the model. She clearly values that. She showcases that and demonstrates that in everything that she does, and it does have an impact on all of us. So collaboration, right? Dividing and conquering, it just makes things a little bit easier. Tiny Earth, leverage the network. What is publishable, collaboration, journal selection? Again, the things that others are talking about and will continue to talk about today. And as far as the buy-in piece, we now have an opportunity at our institution, because of a change in administration as a unit, as a program in human biology, to articulate what scholarship looks like and what it should look like, given what we do as a program in human biology, as faculty that belong to that program. But ultimately, it does have to go through the dean. And so the dean has his own ideas about what scholarship should look like. So, you know, I think on the one hand, some of these things may appear simple, but they can be very challenging. So as far as institutional buy-in, with the time I have left here, yeah, one thing I will get into with this, I've been at UW Green Bay now, I think this is my 27th year that I'm finishing. And in human biology, there has been a lot of, during that length of time, what I've seen is, well, the rest of the institution just doesn't understand biomedical research. And what I would recommend is, instead of approaching things that way, although that is true, I think we can go boldly. And one way you can go boldly with this is, as a strategy, thinking about buy-in, is really connect what you're doing to mission statements and strategic priorities. And it's amazing, as simple as that may seem to do, as obvious as that may seem to do, we haven't done that. At least during my length of time, which is 27 years, it's been mostly, they don't get it, administration doesn't get it, et cetera, et cetera, et cetera. And it's, you know, we always feel better, all of us, when we feel empowered. You know, one of the worst things that we can all go through is to feel helpless, right? So what we are doing now, and everything that we're doing in this way to articulate our message for what scholarship looks like for human biology, is we're really, really focusing on mission statements, strategic priorities, and all those kinds of things. So thank you for all your time. And I am happy, next slide, to pass things off to my good friend and colleague, Dr. Angelo Kolakithis. So thank you.
Brian. So if we can go on to the next slide. So I'm Angelo. I work over at NWTC, which is a very small tech college. And we have traditionally, as Brian was saying, very high teaching loads. When I first came here, we had no science program whatsoever. And so since we have no science program, getting institutional buy-in to do anything related to science was a very, very tall order. Likewise, NWTC is also a commuter technical college, and there's a high push to keep students off campus. And so for our lab time, we get, for microbiology, for lab, we have 1 day a week for an hour and 50 minutes. So just to paint the picture here. But we're still able to run most of the tiny Earth experiments. And what we're able to do is get through the phenotypic and genotypic testing. We go through all of that. We have time for PCR. But for the extractions, typically we don't get to that in the semester. But one time we had a capstone student in the newly formed science program that I helped create, who was interested in taking some of these really promising isolates and trying to extract the secondary metabolites and then testing those for activity. But she was also interested not only in whether or not those were bioactive against the escape pathogens, but whether or not they were toxic at all. Because you can imagine that we can be testing isolates all day for antibiotic activity, but if they end up being toxic to human cells as well, that's not a good candidate. And she tested a bunch and she found one that was not cytotoxic against the human cell line, the HeLa cell line. And so I thought this was really cool results. She did a poster presentation. She wrote this up really well. And I thought it might be publishable. But of course, I was hesitant because being at a technical college, there's a lot of pressures that I see and feel every day about tech colleges and tech college students in general being less than. And because, if you're going to an open access tech college, you might not be, worthy of the scholarship of the university. And so it might be perceived as, well, any research work coming out of a tech college is being done by students that are not real, quote unquote, college students. Not only that, there's no budget for publishing science, let alone budget for doing science. And you could imagine publication fees would add up. And then since we are a commuter college, making sure that I still have regular contact with students after class. If you know anything about students, after they're done with their class, you might not ever hear from them again. So next slide. So not knowing, what to do here, I wanted to try to figure out, well, are there any journals that are student friendly? So I went to the best place for information ever, Facebook. And I went on the TEPI Facebook page and kind of just threw out the question, hey guys, anybody ever publish some of this work in a student-friendly journal? And Nicole, many of you guys know Nicole, kind of gave me some options. Micropublications, JMBE, Microbiology Spectrum, FindFocus, all of these possibilities. And so if we can go on to the next slide, I started looking up some of these journals, and I decided to initially put together the article for Microbiology Spectrum, and it was initially rejected from there for reasons of being a lot biochemistry based, because we were talking about toxicity a lot, not enough microbiology. And so then we went on to looking at other publication methods. And so I started reviewing the Micropublication Biology Journal. And it's a very interesting model because they limit you to one figure. Now, if you know anything about science, we generate lots of figures, but there's a little asterisk to that because you can put, panels A through W if you want, and be able to do it like that. And so they asked me to, because I initially didn't read the directions too closely about that one figure rule. So they asked me to very nicely break it down into certain sections and have one main figure per section. And it's interesting how they do this because, it's one story, how we like to present research. We have iterations of the experiments and they all connect to a story. And the way they put together those stories is that they say, okay, they're separate publications, but what we'll do is we'll link them all together in this piece called integration. And so the editors at this journal were really responsive to any questions that I had, really responsive throughout the peer review process. Everything is traditionally peer reviewed. And the cost is about $250 per article, which if you know anything about publication costs, that's really, really low. It used to be free. This was a grant project. And so now that they're charging $250 per article, they also say, you know, if you really can't afford that, we'll waive the fee. So there's waivers available. I also sometimes publish in JMBE. They have a bunch of different article types. 750 for some article types. That's a little bit more expensive for other article types, but like other journals, they also have a fee waiver available. And as somebody who has a zero budget, we're doing science at all, having those available fee waivers are really important, because lots of times when we're trying to think about whether or not we should publish ourselves or publish with our students, right? Budget dollars are hard to come by, especially in institutions that don't know or appreciate science whatsoever. And so we were able to publish a little bit. So go ahead and go to the other slide. Here we have a few articles. The antimicrobial activity, toxicity of secondary metabolites, time course of the secondary metabolites, and things like that. That's all my two students who wrote those up. And then I've also published on how to bring undergraduate research to community and technical colleges, how they'll spread. And then recently, Brian and me and a few colleagues published on the Tiny Earth and Tidal Town conference that we all put on together, which is involving the community and addressing the antibiotic crisis. And so these are all articles that had something to do with the Tiny Earth work that we did. And when it comes to institutional buy-in, if we can go to the next slide, it turns out when you publish something at a technical college with zero budget, it gets attention. And so we had a lot of attention brought on by the antibiotic research that we've shown over the years. It's kind of a little niche product, if you will, to say, hey, we're doing science at a research level that's publishable at a tech college with very little to no budget. And this gets the attention of news media. And then when the news media comes and writes all these stories, then presidents and vice presidents and deans start to pay attention and go, oh, Angelo, so what do you guys need again? How can we help? And my under the breath comment was, well, you could have been helping this whole time, but yeah, I'll take any sort of budget that you have. And so that's kind of the way I try to garner buy-in. I do everything as low budget and free as I can, and then get the attention of the community to show that this is a good thing and that we should be given resources to it. And then you can go with the old saying, if you build it, they will come. And then next slide, I would like to introduce Jody, who's at Luther College, is going to talk about her work with non-traditional dissemination of science.
Thank you so much for that intro. Can you hear me okay? Just checking. I've been having some tech problems here, but I think I've got them resolved. So yes, thanks for that intro. A non-traditional way of starting here is just to elevate for all of us a reminder of like goals of science. And so if you can do the next click, and this is something we're all familiar with, building knowledge and understanding of how the world works. We have a method to do that, and then to use in applications this knowledge to devise solutions to specific problems. So this is, essential, foundational. and I would argue perhaps insufficient to be in our repertoire of approach. And so if you could highlight, so I want to highlight what I think is a limitation of the approach. And it's perhaps best captured by this quote from Brené Brown. So we love to believe we are rational, thinking people who on occasion feel emotion. We are not. In fact, we are emotional feeling beings who on occasion think. And so there are loads of things coming out in the neurobiology and neuroscience world that are pointing to the human emotion and heart connection to mind as being foundational for human decision making. And so we are human beings that are scientists. We are emotional human beings that are scientists. And we live in a world where emotion is tied to human decision making, which is absolutely connected to the application component of science. So these are the pieces that I have that turn into drivers for me. So next slide, please. So it's led me into different directions to try to disseminate information. And I'm going to talk about two of them today. One of them is a TEDx talk, and the other one is a collaboration with artists. And the TEDx talk came up as an opportunity last spring. And I'm not going to go into all of the details here, but I will happily engage with folks who are interested in learning more details about this. But key things that I want to emphasize: at Luther College, students are the key. They do it all. They're the ones that have the connection and sought the license application with TED. And they run it, they solicit the proposals, they decide who's speaking, they run the conference, and they video it, and they were the drivers in the editing, although I was a heavy consult in that. So they chose my talk, and in fact, I gave them 3 proposals, and they chose very much, they were unanimous in their choice on the one that ended up getting delivered. And so it's called Power of the Unseen, and a quick synopsis is, it's a social justice for the microbes and women landowners sort of talk. So the power, trying to highlight the power of the unseen soil microbes as foundational for life, and the soil crisis that's been happening and continues to, with the power of women landowners to save them. And so this has been very much inspired and influenced by Jo Handelsman's, I mean, that's really what kind of started me on the idea, her presentations, her book, but also my mother, Rick Cruz, at ISU. And I'll just highlight that this was a big deal in my mind if I was going to do it, because this lives in the public sphere, which is both scary and an opportunity, but it lives in the public sphere forever in the TEDx realm. And so I invested. And so I want to just zoom down here on the challenges and opportunities. So I invested a lot of time and a lot of research, both like literature research, but also human research, meeting with people for both the content and how to do this. And sources and crediting is just elevated very high. The final product needing to be a quality that you're happy with is also very high. So there's investment and effort required, and I want to share from what has happened for me, completely worth it. And I would encourage anyone and everyone to consider this as a way to disseminate information. So it's created so many opportunities, but the best way that I can say what happened to me during the process, research and delivery, was it lit a fire. As I realized like where we're at with soil and the potential for this idea. So if you could move to the next slide. These are some of the outcomes of this fire that was lit by doing the TEDx talk. So, and these connect with each other. So the collaboration and partnerships and grants have influenced more presentation requests. So versions of this talk have gone multiple places locally and regionally. The 3 in blue are requests I've gotten in the last couple of weeks that I'm trying to decide if I have the bandwidth to do. But I'll highlight one of the, so I did a version of it at the Iowa Water Conference last fall, and someone at that talk came up to me and said, I want to do something big with this. I want to do something big. So when I heard, like, connecting to the previous speaker, if you build it, they will come. This person has led a whole team to do a Women in Soil event at the Iowa Field of Dreams. And they asked me to headline it, but it's not like, now it's turned into like, there's a soil food web. So many partners coming in. It also involves a finale that's tied to a collaborative work with the arts that I'm doing. But this is getting, as the previous speaker also this led by like our rock star, like women team, planning team. This is going to be on, they've got $11,000 worth of donated news coverage on KCRG elements. They've got radio involved in it. Like, it's gone places that I wouldn't have imagined and that I don't have the bandwidth to create. So other things, so we're really excited about that. Other things here, though, are also really important. It's led to a lot of different partnerships, and some of those partnerships led to so I turned around immediately within the next week and wrote a grant, basically said my TED talk. And then that led to, with partnership, a much bigger opportunity for a grant, which has now enabled the next 4 years of research projects for Luther students on soil health, looking at the biology of soil. And it's also it's in the process of leading to teaching innovations. It's adding a new opportunity for collaboration with artists, and the grant has enabled a full year sabbatical. And so, like, all this came out of the research and delivery of a TED talk. So for me, it's really turned into something very significant. Next slide, please. And so now I want to this is my last slide, but I wanted to highlight that I also, this is not a new thing, but Soul of Soil is connected and coming out of the TED Talk. I collaborate with artists at Luther, and I have found this to be one of the richest experiences of my academic life. And so in 2015, we did Body of Water. So I've highlighted Jane Hawley, dance professor. So she's my primary collaborator. But then, of course, like all these other artists who are also faculty members in different areas of arts at the Luther are pulled in, and it's this huge collaboration to put on a performance. And so this Body of Water went further than Luther. So we took it to Iowa State, Grinnell College, and some other places. It resulted, so that project and the process led to invitation to do a book chapter in this book, Extraordinary Partnerships. So Jane and I had that. And then I had dreamed of a sequel, and then inspired by the stuff going on with the TED Talk. And we're doing that in 3 weeks. And this one is one that, so Body of Water, I was in as a narrator and as a video, in video form. And I told Jane I would be brave enough to be in the next one live. And so this is definitely taking me out of my comfort zone. However, what I will share, and this ties back to the very beginning of my presentation, I can give a long PowerPoint that is just jam-packed full of really good data that is really provocative in terms of needs in the world. But that I've never caused someone to cry over a PowerPoint packed full of data. And that has and is happening with both of these performances. And so it is, and people remember, your memory is tied back to what you have experienced emotionally. So people come up to me 10 years after a Body of Water, and they can describe a scene with great detail and how it affected them. So I want to just highlight, that is the driver for human decision making. And if we seek to have applications of our science, to maximize those applications, I would say, I think it's critical that we include this component. And then the final thing that I will share here is that, as I'm highlighting here, combining the arts and the sciences is just like our soil organisms and our relationship with soil. If it's gonna be healthy, it's built on reciprocity, and both entities need to benefit. And so I've already shared with you some of the ways that the sciences have benefited, like the reach that these performances are the ones that sell out in our arts department quickly and have the broadest, most diverse audience members, because farmers are coming. And so the arts are benefiting, as well as so we are more than the sum of our parts. So we're accomplishing something bigger than the individual components. And the arts are undervalued and underappreciated and underfunded. And this gives them access to material that they don't have access to. And they're so interested in collaborating. And the students are on fire, because they see what they're doing matters, and because it's combining these things. So I'll stop there, but I consider, very happy to be involved with those pieces. Thank you.
Thank you, everybody, for all of your amazing work and contributions to both this workshop and beyond. We have about 14 minutes here for a quick Q&A. If you want to see the resources that these folks have gathered for you all, I have a link here. It's a collaborative document, so if you have your own tips and tricks, feel free to add those to the document so they have a living place. Feel free to raise your hand to ask a question, or you can just put something in the chat too. Poonam S has a question in the chat. Maybe this one's for Angelo. Asking for guidance on effective extraction methods of antibiotics from isolates.
Yeah, so we used both the methanol extraction and the ethyl acetate extraction that's listed in the Tiny Earth book, which is basically you grow your isolate on an LB plate, you make a lawn, you take a scalpel, chop it up. We use little tiny mason jars to put it in. We throw it in the freezer. Depending on the time, we leave it overnight. I just want to make sure that we get good freezing. And then we either put 15 mils of methanol in, or if we're doing the ethyl acetate, it's a mixture of, I believe, 15 mils of ethyl acetate, 10 mils of water, but don't quote me on that. I don't have it in front of me, but we have found that the ethyl acetate seems to work a little bit better in our hands. And so then after we take the ethyl acetate out and put it into another jar, we go put it in the hood, let it evaporate, and then we reconstitute with methanol, depending on what we have the vials pre-weighed, and then we typically do like a 1 mg per mil concentration. Take that, put it onto another plate, 10, 20, 30 microliters, overlay it with whatever escape we're testing, and then we look for zones of clearance.
Thanks, Angelo. Laura Pickle has a question about permissions and authorship with Tiny Earth, and if the folks need to run ideas by Tiny Earth, for example. We love working with you all on expanding your ideas and helping you. We don't have any hard and fast rules. Is Sarah on the call? Sarah? You mind answering that question?
Yeah, I'll echo what you just said. We're always happy to just be a sounding board too. Sometimes that's just the best place to start because we can be aware. That also helps us if like there's a webinar and we're like, oh, somebody, so-and-so could talk about these things. So for example, all the folks here today, just folks who've, talked to us and reached out at some point. There are many more of you who have done other publications or TED Talks and things, and we recognize that too. So yeah, there's no hard and fast rules. That said, I think the place where it gets tricky is if, for example, you were going to publish a student research guide that looked and felt like the Tiny Earth student research guide, that's a very different conversation. But if you're publishing on your student research, you're publishing on your study that you did on your classroom, no, that is all you, that is all yours. Go for it. I strongly encourage anyone to just give credit and attribution to Tiny Earth, the Tiny Earth Network, and the acknowledgements. But unless you're lifting content and just follow general academic rules, go for it.
Mm-hmm.
Can I just go back up? There was, Kristen added a comment in response to the extraction question and put a link in the chat that followed up on what Angelo mentioned as well with some expanded ideas for protocols. So just so folks know, Kristen's been working with the TEPI training group to help really hone that chemical extraction process. Do you want to add anything to that, Kristen?
Not really. There seemed to be a lot of questions about it, so I made that protocol. It should be flexible. That's one way to do it. I tried to include lots of pictures. Reach out with any questions if you have them.
Great. Thank you both. With regards to advertising COPs, there's not a page yet, but I've been wanting to make one. And so you could probably expect something like that pop up this summer, if not in the next couple of weeks. I'm actually working on editing both the Tiny Earth and the TEPI website, so keep your eyes peeled. And as always, just stay tuned in the newsletter. There's always that's mainly how we've been connecting TEPIs and recruiting folks to these type of things is, hey, in the newsletter, join this thing or check this out. So, or the TEPI website, of course. We also post everything, pretty much everything that goes in the newsletter lives on the TEPI website. Any other questions? Happy to answer anything. Brian had to leave, unfortunately. He had another commitment. But the question from Jody is, how would publication with artists such as in performances, in books, etc., be viewed at your institution?
I can answer for my institution since we don't have a formal arts community. It would be kind of something that would be a collaborative effort on the side. I'm not sure how what the amount of pushback and blowback I've had at the whole publishing of sciences, if that would be palatable for them. I think it would be if it didn't cost any budget and I was doing it on my own time. But if it got good news media feedback for the college, I think that's where they would change their minds.
If I could respond, thanks for that. I would say, and I'm looking at the responses, I think that there's a challenge, I feel a challenge from the Scientific Academy about whether that is valid. And I'm tied it to Brian's message and questions about that as well. I think where I'm working with artists, for example, like that is their scholarly work. That's their scholarly work, but they don't have access to it unless I'm involved. But is it counted by even my own department or institution as scholarly work for me? And I'd like to challenge the paradigm of maybe what science is willing to consider. Thank you and I invite anyone else. I'm just really curious about that at other institutions.
All right. Sarah has posed an interesting question in the chat for the audience, so I'd love to invite the rest of you to come and share some of your thoughts. Sarah asked, has anyone on this call published on Tiny Earth or had your students publish their findings? And what would you be interested to publish? Folks have ideas on that.
To any education research as well. It doesn't have to just be Tiny Earth. And I'm looking at some of you because I know some of you have published in this space.
Marisa? Marisa, did you want to share? Or you just raising your hand to say yes?
Yes, hi. Good morning, everyone. So I don't know if you can call this publication necessarily, but I'm gonna present a workshop regarding Tiny Earth in ABLE conference this summer. And as part of that, they basically publish what you do for a workshop. And I mean, that's a way to go about it, I think. I mean, I'd like to hear your take on it.
Can you say a little more about it?
Several of my students try to modify some of the procedure. For example, they try to use fruit juice in their media to see if that cause more antibiotic production from bacteria, and they had some limited success, but I think it's a way to go about modifying all these procedures that we have, and I'm trying to present that as a again, a lab technique in this conference. But as part of that, they publish all the proceedings. So what do you think, Sarah?
Yeah, no, I think I missed the first part of your question. So are you asking if that is something what's your question?
So I don't know if this is called publication necessarily, but I think, little by little, starting from places like this, people can expand their work. So just an idea.
Yeah, no, okay, thank you. Now I got all the pieces. So I think, depending on how you frame it and where it goes, certainly publication. I think another piece of the puzzle here that we didn't really talk about is the value of presenting things, right? Presenting things at conferences, even if it's not a paper per se, is valuable too, right? That's one more line in your resume, especially if it's something invited. There is a place, I know, Angelo gave an example of where, his students can publish, their research findings. I don't know if they could do methods too, but there is something also called QUBES, QUBES. I don't know if you're all familiar with this group, and for the life of me, I can't remember what the acronym stands for. But it is a group of STEM educators who have it is a giant community of practice. Adam was talking about communities of practice earlier, of folks who share out their curricular ideas, usually with, tagged on with assessment and evidence-based practice practices. I don't know that there's always if you have to have assessment, but it is a place to share out things like that too. So thank you, Adam, Justin, for pasting that in there. If any of you who are CUBES users want to add to that space, that can be a really, really nice tool for folks, and so community driven.
And if I can add two things in that resource document, if I may, because again, I'm very scrappy and trying to get as many resources as possible, but many of you might not know this, but NEB and New England Biolabs has this thing where you can write to them and get $1,000 in free reagents to do your PCR or any other experiments that you do, just for writing that as $1,000 per year of free stuff. And that's really carried us through a lot of our genotypic work here. And then another thing for journals beyond what we shared here today, the Council for Undergraduate Research has a running list of undergraduate research journals, of which there's, gosh, I don't know, probably 100 or something like that are all listed there. So there's not a lack of space in this way with publishing with undergraduate. There's all sorts of things out there. The harder part's going to be making the choice of which one to choose. So it's not for a lack of choices. There's lots of choices. You just have to think about where and when and why and how.
Great. Thank you everyone for this. I see Arthi put a chat in there, comment in the chat, and I'll just say this and we can close, but we published during the pandemic on our pivot to online. We published on the eGEDA materials we created too. And so sometimes it is a community of practice within our group, the community that Adam's got going. So sometimes it's just, finding folks within this particular group to move things forward and publish together too. Thank you for bringing this all together, team. This was really informative and useful. I loved seeing all of these together, and we'll see you all at the symposium this summer. Register soon and bring all your students. June 12th and 13th. Can't wait to see you all. Bye.
Bye, everybody. Thank you so much.